Dear student, Assalamu alaikum. My name is Dr. Muslim Khan, Assistant Professor, Department of Chemistry, KUST. So today we will discuss hyphenated technique. Course code is CHEM453, mass spectrometric and thermal method of analysis. One of the most important hyphenated technique when high performance liquid chromatography means HPLC is connected to the mass spectrometer, which is known as HPLC-MS. This is a hyphen in between the two techniques. As two techniques are basically connected, so that's why it is known as hyphenated technique. Nowadays, it is called coupled techniques. But before going into the connection of HPLC with MS, we will discuss first of all HPLC so that it makes your knowledge about HPLC a little bit fresh. Also, we will discuss MS, and then in our next lecture, we will discuss the combination of these two techniques, that how it take place. So first of all, HPLC. How liquid chromatography change into high performance liquid chromatography. First of all, in chromatography, two things are needed. One is this high degree of separation, and the second one is reduction of analysis time. So for high degree of separation, the column material, in which column is made of refinement of a packing material, was needed. So that packing material was refined to in between 3 to 10 micrometer. But to refine the packing material, then it is very difficult to pass the mobile phase through this column, which is having a very small plate or packing material size. For that one, you need a high pressure pump, which can pump this mobile system by a pump. So delivery of effluent by a pump was needed. So for that thing, a special equipment was the demand of the time that can withstand the high pressure. That's how the liquid chromatography change into a high performance liquid chromatography. That's how the arrival of a high performance liquid chromatography take place. What is high performance liquid chromatography? HPLC is a form of liquid chromatography used to separate compounds that are dissolved in a solution. HPLC instrument consists of a reservoir of a mobile phase, a pump which can pump the mobile phase, an injector in which sample is injected into the mobile phase, and then a separation column which can separate the analyte from each other, and at last a detector. So compounds are separated on the chromatographic column by first when injecting a sample mixture onto the column. The different component and the mixture pass through the column at different rates due to differences in their partitioning behavior or interacting behavior between the mobile phase and the stationary phase. The mobile phase must be degassed before going into the column to eliminate the formation of air bubbles, because the air bubble can damage these packing material which are packed so it can affect the efficiency of the column. Here is the flow chart of HPLC system. HPLC system having solvents reservoir system, which is having either 2, 4 solvent system. So solvent from this solvent system is going into the mixing vial, but before mixing these solvent with each other, it must be degassed by a degasser. If there is a little bit of gases present in the solvent, it must be degassed to eliminate the air bubble formed in the column. Once these solvent systems are mixed with each other, it is pumped by a high pressure pump to the column. But in HPLC system, most of the time people are using pre-column so that the unwanted things are basically guarded by this column. So that's why this column is also called a guard column. So a pre-column is fitted before the actual analytical column. So after this, the mobile phase, when passed to the guard column or free column, there is a flow splitter, because all of the mobile phase, you are not allowing it into the analytical column. So flow splitter is splitting the flow most of the time 5 to 95% or 10 to 90%. It means that only 10% of your mobile phase is going through the column while the rest is going to the waste. So here a waste bin is also there which can collect the extra uh, amount of a mobile phase. After this, a sample is injected into the path of this mobile phase. So mobile phase along with sample, what you want to separate, is going through the analytical column where actual separation take place. So column is the heart of the HPLC system where this separation column, and then all the sample along with mobile phase is going into the detector where it can be detected. So there are basically four types of liquid chromatography occur within this analytical column. One is this partition chromatography and adsorption or liquid solid chromatography, and ion exchange chromatography, and size exclusion or gel chromatography. This is how typical HPLC system look like. HPLC system is having this solvent system, these bottles you can see, and then solvent delivery system. Here the pumps are basically fitted. Before the pumps, degasser is also fitted, which can degas the mobile phase to prevent the air bubble in the column. And then an injector system. Here is this automated injector system which can pick the vial and inject the sample into the path of this mobile phase. Before this injector system, here a splitter which can split the mobile phase 90% to the waste and 10 or 5% into the column. So sample is injected in the path of this mobile phase, and then sample along with mobile phase. Here in this system, this part of the chromatographic system, a column is fitted So a column is fitted when the sample along with mobile phase is pumped through this column by this high performance system. And last detector, but we will maybe in this lecture we will only mention it but not discussing detector, because in hyphenated system this detector is basically deactivated, and this HPLC system is directly connected to the mass spectrometer here. So means mass spectrometer is acting as a detector and hyphenated system, and it lost the waste collector and data collector, means recorder, is there in the HPLC system. So the uses of HPLC. This technique is used for chemistry and biochemistry research, analyzing complex mixture, purifying chemical compounds, developing process for synthesizing chemical compounds, drugs, isolating natural product, predicting physical properties. It is also used in a quality control to ensure the purity of raw material, to control and improve process yield, to quantify assay of final product, or to evaluate the product stability and monitor degradation. In addition to these uses of HPLC system, it is used for analyzing air and water pollutant, for monitoring material that may jeopardize occupational safety or health, and for monitoring pesticides level in the environment. Federal state regulatory agency use HPLC to survey food and drug product for identifying confiscated narcotics or to check for adherence to label claims. So the function of the injector is to place the sample into the high pressure flow and narrow volume is possible, so that the sample enters the column as a homogeneous low volume plug to minimize spreading of injected volume during transport to the column. The shortest possible length of a tubing should be used from injector to the column. When injection is started, an air equator rotates the wall, solvent goes directly to the column, and injector needle is connected to the syringe. The air pressure left the needle and the vial is moved into the position beneath the needle. Then the needle is lowered to the vial. The column, coming to the column of the HPLC system, the column is one of the most important component of HPLC chromatography, because the separation of the sample component is achieved when this component pass through the column. The high performance liquid chromatography is made out of stainless steel tubes with diameter of 3 to 5 millimeter and a length ranging from 10 to 30 centimeter. Normally, columns are filled with silica gel because its particle shape, surface properties, and pore structure help to get a good separation. Silica gel. Silica is wetted by nearly every potential mobile phase, is inert to most of the compound, and has a high surface activity which can be modified easily with water and other agents. Silica can be used to separate a wide variety of chemical compounds, and its chromatographic behavior is generally predictable and reproducible. This is the picture of a typical HPLC column. As I mentioned it, so the typical HPLC column made of a stainless steel. Inside this stainless steel, a packing material is basically loaded, usually silica, uh, sometimes polymeric bone column. So this is the column how it looks like. What are the parameters that can affect the system? That is column material. What type of column material is inside there? Deactivations, stationary phase, and coating material, which can affect the column parameters. And also instrument parameter can affect the system as well, like temperature. At a different temperature, the peak shape, the resolution of the peak, etc. is they're different. The flow is also very important. Signal and sample sensitivity. Some of the samples are very sensitive to some of the material, column material, and also the flow of the system and the detector. Sample parameter can also affect the system, like concentration of the sample, what type of matrix sample is in it can affect the sample system. And solvent can also, what type of solvent you are using, methanol or acetonitrile, or what type of buffer you are using can affect the system. And sample can also affect the parameter. There are several types of chromatographic column. Depends. So that's why different types of chromatography are there. Normal phase chromatography, reverse phase chromatography, size exclusion chromatography, ion exchange chromatography. So first of all, the normal phase. This is just to refresh your knowledge about chromatography, because we are connecting this chromatography into the mass spectrometer. In this column, the type, the retention is governed by the interaction of the polar part of the stationary phase and solute. For retention to occur in a normal phase, the packing must be more, stationary phase must be more polar than the mobile phase with respect to the sample. While in reverse chromatography, this is reverse, the stationary phase must be less polar. So in this column, the packing material is relatively non-polar and the solvent is polar with respect to the sample. Retention is the result of interaction of a non-polar component of the solute and non-polar stationary phase. Typically, stationary phase are non-polar hydrocarbon, waxy liquid or bonded hydrocarbons such as C18, C8, and the solvent are non-polar aqueous organic mixtures such as methanol water, acetonitrile water, etc. While the size exclusion chromatography, the HPLC column is consists of substances which have controlled pore size and is able to be filtered in ordinarily phase according to its molecular size. Small molecule penetrate into the pore within the packing, while larger molecule only partially penetrate the pores. The larger molecule elude before the smaller molecule. This is the whole theme behind size exclusion chromatography. While in ion exchange chromatography, the sample components are separated based upon attractive ionic forces between molecule carrying charge group of opposite charge to those charges on the stationary phase. Separation are made between a polar mobile liquid, usually water containing salt or small amount of alcohol, and a stationary phase containing either acidic or basic fixed site. Uh, we are not discussing this selectivity factor. We have discussed it before in our previous lecture. So there are different types of detector. Even we are not discussing these detector, because in hyphenated techniques, anyway, we are making these detector inactive, deactivated. There are different types of detector like IR, fluorescence, refractive index detector, but we will only focus on a mass spectrometric detector, because we are connecting this HPLC system to the mass spectrometry detector while its own detector is being deactivated and this hyphenated techniques. Evaluation parameters for chromatography are like efficiency, resolution, alertness, retention index, and column bleed, capacity factor, etc. So for these books, for this lecture, you can use J.D. Christian Analytical Chemistry, Scooge and West, Fundamental of Analytical Chemistry. Also, you can use J.D. Christian Instrumental Analysis for this one. And thank you very much.